we'd like to start by asking you, how should a researcher pick a question to work on? For instance, how did you start working on fly larval segment patterns?
My question was not the segment pattern, but my first initial question was how to identify morphogens. And at the time, morphogens were sort of a miracle, and one didn't really, they were very elusive, one didn't know anything about them, and they were postulated on the basis of transplantation experiments in frogs and in other instances. And gradient models have been proposed in order to make, from a simple uniform pattern to get more complex patterns, depending on the concentration of one particular substance. But no gradient molecule had been identified. And I wanted to find them not by transplantation experiments, because they had failed over several decades actually. And I realized that genetics would probably be the approach to identify morphogens, because they must be somehow encoded by the genome, and you may be able to make mutations. And when you have a mutation, then you can identify it and eventually also isolate it. And at the time, the best model organism to do that was Drosophila, maybe the only organism where you could do that. The nematodes do not have eggs which are laid, and you cannot manipulate them. And I didn't know about them actually. So one knew about mollusks, and one knew about Hydra, and one knew about many other organisms, but only Drosophila you could do genetics. And this was the reason. So I set out to identify morphogens. And then it turned out that the genetics to get maternal mutants is really very, very difficult. And we first then set out to do the zygotic mutants. And there the segmentation, the patterning of the embryo was very striking and led us to first analyze this. But later we came to the morphogens as well.
During this whole time, when did you realize that you were going to find something really fantastic? Like, which was the Eureka moment for you in this?
I had several actually.
Okay.
Yeah, when I started working with flies, it was extremely exciting, actually. There had been this one mutant which had been described in the literature, which had two abdominal ends bicaudal, and I found it in an old stock. And this was a big excitement. And then I found a new mutant. In my first screen, I found a mutant which I called dorsal, which is responsible for the gradient along the dorsoventral axis. I found that within the first half year of working with flies, and I was extremely excited about it, because it was clear that this was, although at that moment the interpretation was not there, but it was clear that this was a very, very, very important and the first real good maternal mutant. And when we did the segmentation screen, we found these parul patterns and the gap genes. This was also very exciting. And hedgehog with his,
But did you immediately realize the significance of these? Because you see the, you can see them.
Yeah. I don't know whether you need a special nose, but it's true that some people don't see the things, even if it's before their eyes. You must, in order to make a discovery, you must recognize that you made a discovery, and you have to recognize the significance of the finding. And we were well prepared for that. Yeah.
And what about the field? How quickly did they get excited about it?
The field was slow. And actually we suffered for some time from the total lack of recognition. So you tell someone you have an embryo which doesn't make gastrulation, and isn't this exciting? And people couldn't, they had different questions, They couldn't use genetics. They couldn't bridge the gap between a molecule, morphogen, the biochemical molecules, and the genetics. This was very hard for many people. And cell biologists for a long time didn't grasp the significance of our findings, because they couldn't use genetics, being used to just grinding up cells and isolating proteins and maybe mixing them and seeing effects. But they couldn't, genetics is an abstract approach, and you have to deduce from the phenotype what's going on biochemically. And this is a step which is difficult to take. And we didn't take this step actually. We just looked at the phenotypes and classified them, and the biochemistry came later. Yeah. And we didn't do that ourselves initially.
Yeah. But was that disappointing that you thought that it was really exciting, but people did not recognize how exciting
Yeah,
it was?
we had enough people to recognize it, and we did publish well. So we could publish in Nature, and people, some at least, got excited. But compared to now, I mean, we got very few quotations, because there was no community yet.
So I think many people would also like to know, apart from the great discoveries that you've made over the long course of your research, did you ever hit any major roadblocks at any point in your research where you felt that maybe this question is not really worth following, and how did you deal with them?
Oh, this always happens. You know, you have to span a broad range of projects, and with the risk that some might fail. I mean, it's really good to think hard before you start a project, whether it might be successful and what will come out if everything goes right. This is very important for young researchers. They often don't think far enough. They start and say, I can do this, oh, I can do this, and let's do this and that. So they start from the given experiment or from their small result they have already and continue there. But they should, I think it's really a wise thing to think about the end result. What if everything goes right? Can I anticipate what will be the finding at the end? And if this finding is dull and not really interesting, then you should stop at the beginning, yeah, and should rather look something where you have a very interesting problem, which has a very interesting, may have a very interesting answer. If the answer is not interesting, you might not do the project.
But how far into the problem do you know that?
Well, sometimes you have failures. I mean, one classical example is when you, in our field, you identify genes by mutations which cause interesting phenotypes. And then you clone the gene. And this is something which had been very difficult initially. Now it's much easier, but initially it was difficult, and also now sometimes you cannot do it. And sometimes you find a protein which does not tell you anything about the process. And there is also no real means to find out. In many cases you find, say you find a transcription factor and you can do an in situ, and then you see, aha, great, it's expressed where I expect it to be expressed. If you find an enzyme with unknown function, and no one has ever worked on it, sometimes you just should drop it and start something fresh. This is very frustrating, but sometimes with the most pretty phenotypes, you do not succeed in finding. This is one example where you, I tend to advise people in the lab to drop the project. And also, in some cases, you have mutations which are just very weird, messing up things, but are not really crucial for the core process. And this is something where I also would say, hands off. If it gets too complicated, don't do it.
So you have a really long career. So every few years, how do you keep reinventing yourself? Because that's very important too.
Well, I'm very curious. So I look around, and when often findings or results provide a new step to a new question. And I have also the habit of, when I have collaborators, especially postdocs, that when they do a project in my lab, they may choose as much as they want their own project within a certain limit, of course, and they choose projects which are in the core of the entire research done in the lab. But then at the end, usually they take the project with them. So I do not continue on the same lines in most cases, only for the students' projects. Because the students I advise to go away and do something different. Sometimes they don't do something different, sometimes they take their things with you. There are exceptions, but in general, whenever someone has done a good project in the lab, they take the project with them. And I do not compete with my ex-postdocs, usually. I collaborate perhaps sometimes, but I do not compete, and we start something new. And actually, when you do genetics, you have so many things which are exciting, and actually to choose the right ones is and not do others is sometimes very difficult because everything is nice. And some people in the lab fall into this trap that they do too many things because everything is there and they can clone this and they can clone that and they can clone that, and they try, and then it doesn't get done. And so this is tricky, and I have to guide the people in this respect to keep focused on something where they can get an answer.
Where do you think that Indian science, particularly basic research in biology, stands in the world, and how do you think can it be improved further?
I think there is wonderful Indian science, and we have in Europe we have many Indian researchers, graduate students and postdocs who participate in our science, and they're just as well trained as the European scientists. And we have to select them, of course. I mean, India is a big country, has many universities, and not everyone is perfect. But we have these graduate programs, and we have very good students from India. And when I look around here, I mean, the institutes I have seen are excellent. I mean, no question, same quality as our institute. I mean, different because we have better connections, perhaps, and we have richer resources or so, and easier access to things, I think. But this is getting easier here too, I think.
Gradually, yes.
So, as a woman, did you face any difficulties when you started?
Oh, sure. Well, I was starting my career at a time where I was really always a big exception. You know, one of 200 directors in the Max Planck Society, things like that. People were not used to dealing with women. Many people, especially the older ones, just had a very strange attitude, and they sort of tried to flirt, or you know, it was not so good. And actually, because there were so few women, discrimination was not an issue. It was not discussed because there was no community of women to discuss it with. So I was lonely, and in many cases I just got treated really not very well, and I had to face it, and I had to go through it, and I managed, you know, because I was very strongly committed to doing my science and not give up. But there were times when I was really desperate, I must say, because I didn't get much support and help, actually. But
so even today there aren't so many faculty who are women. I mean, as a ratio to the number of men that there are.
I think it's fair. It's much better than.
Of course, much better, but still not equal.
It needn't be
not
equal.
Even close to equal.
I don't think it need to be equal. I don't think this is a good aim to have it equal, because many women choose other professions, and many women don't want to focus as much as I did on. I mean, I really concentrated on science, and I didn't have children. I don't have a family. I was very determined, and this is not for everyone. And many women prefer to have maybe richer. I had a rich life. I don't complain, and it's not that I gave up things, on purpose. But I choose to do this, and other women might choose to do more of family or more of cultural things, friends, and this is legitimate. And I think it's not to be expected that women will be as happy as men in these high-class, very competitive professions. And many men who are in leading positions have wives who do nothing but caring for the man and sort of being the secretary and the butler at the same time. And this is something which is also, many women like that.
No, but
and men like it too. So why give up on that?
No, but often they're forced to give up. So is there what can be done to improve this situation?
Well, what do you mean forced to be? No, I mean they have choices. Circumstances might make it very difficult, but this is because maybe they want more than at the present time they can face. I mean, if you want to be an excellent mother and at the same time an excellent researcher, and then a wonderful wife to your husband, and educated and beautiful, this is a lot. You cannot do all these things at the same time. So you have to choose. And then if you choose that you want to be the best mother in the world, you must spend quite some time with your child. And then you cannot spend this time in the laboratory, so your research will suffer. So you have to maybe do a compromise and say, I trust daycares, I trust the school, and I do not have to do all the things with my child personally. And if you do that and you manage to do that, many women have problems there. I have seen good cases where women have done a good career with children and have been quite successful. And you have quite a few women faculty in your institute here.
Yeah, but not as...
I don't know. In Bangalore, I think there were 50% women faculty.
No, not 50%.
Well, but
Maybe
it's,
like,
I mean, 50 is too much,
30
actually.
maybe.
Yeah.
30, I think 30 is fair. Yeah. And more is perhaps pushing, a sign that you push women too much. And this is a danger too. I mean, if you push women in a position where they really have a lot of responsibility and they cannot bear it because they have too many other things, then they fail, and this is also not good. And you also have to keep in mind, research is only fun if you are successful, if you make discoveries. And if you do not have enough time, energy or focus to make discoveries, then it's not a great job. I mean, you may do teaching, okay, but as a researcher, you must be successful. You must make discoveries. And if this is not in the reach, I don't know. I think it's tough.
But we've heard of your foundation. That is a really nice step to helping women cope.
Well, this is teaching them to, what I said before, you cannot do everything. You cannot also do the washing, the laundry, the cleaning of the floor, the scrubbing of the windows and knitting your own sweaters and things like that. You have to have other people helping you. And you have to pay them, of course. They couldn't, I mean, except you have a mother or some relatives who do it. But you have to learn to ask for help from people and pay them for that. And in Germany in particular, maybe here it's different, I don't know. Maybe here labor is cheaper, so it's easier to do.
Everybody has some help.
And in Germany it's expensive, and people are not used to it because they don't want to let other people do the dirty work for them. But they have to learn it, otherwise they cannot be professionals.
So it seems that science nowadays is more driven by applied grants rather than curiosity-driven fundamental questions. Many people think that way these days. So what is your opinion about that?
More translational than...
Translational research is a word which came up in the past 10 years. I had not heard it when I was doing my basic research at the beginning. And it seems to me that there is more focus on that now. And I think there is some attitude of politicians in particular who say, you have promised so much. And people have promised for a long time, if you give us this money, we will solve cancer, we will cure these diseases and so on. And it took a long time, and still the diseases are there and we cannot completely cure them. And it's too much promise, I think. But the politicians now say, no, we want this. And this is in part because also some scientists think we have learned enough now, and we can now, because we can sequence human genomes, we can just get enough information just from doing only research on humans. I think it's a mistake, because you cannot do experiments on humans. You can only look, you can only describe, you can get the genome information compared to the phenotype, but you cannot do experiments. You cannot make humans with particular features. And this requires model organisms. And the word model organism says it. It's a model for humans. The models are better or worse, but in many things we have found out by bacteria. They are also in a way model organisms. They can tell you much about the real nitty-gritty of the cellular details, of the biochemical details. And flies are good model organisms because they have the basic set of genes making the patterns and the pathways. Zebrafish is even maybe better, but more difficult to study because longer generation times, water and bigger sizes and so on. But you need them to study the genes in detail, get at the function.
Do you think there are any special areas on which Indian research should be focused on, or if Indian scientists can benefit the community by working on certain areas?
I think what comes to mind, there are two things. The one is genetic engineering in plants, making good crops which are resistant against insect, pesticide, pests, and you don't have to spray so much. I'm really afraid this country is so rich and is so, hot climate, probably plenty of pests. So your food must be terribly polluted by insecticides.
There's a lot of people moving towards organic food.
But this is much too expensive. And you have, looking at the density of population in your country, you cannot afford this because it requires much more land because the yields are lower. So I think you should really go for genetically modified plants. And this new method, CRISPR-Cas, is so specific that you can do very, very good crops without any danger for human. Even the classical, the traditional genetically modified organisms are fine. We have wonderful tests. The whole United States eat nothing but this food, and they're healthy and kicking. And it's clear that this is a very, very big experiment, and it didn't fail. So you are on the safe side. And I think talking people into eating organic food in India is criminal, because this only a very, very small portion of people, and only the very rich can afford it. And this is very unsocial. And I think this is almost criminal. And talking people into not having GMOs, but rather spray insecticides, I think is also criminal. So I think Greenpeace essentially is criminal, I must say. And also in Germany, in Europe, we don't need that. The country is, the climate is different, and we have a colder climate, we do not have all these incredible pests, and we can pick and choose, and we can also breed conventional, and you use, of course, also conventionally bred crops, but still, it must be, and I heard from cotton that now most of the cotton is BT cotton, and this is really very successful, and has also led to the fact that the less spraying makes the farmers much more healthy. And this is very, very important. And this is a good test case, which you should explore on, and you should advertise, maybe even to the Western world. I think this is really one thing. The other thing, which I find very striking, is biodiversity. You have such a richness of animals and plants here. And this is now not applied research, this is just basic research. You look and see and learn how the ecology goes. There are probably not many untouched areas in India, because it's such a... but
not untouched, there may be tribals living there.
Yeah, but I think it's really important to cultivate the land which is cultivatable, which you can use for agriculture in a sustainable manner as intensely as you can to get the highest yield from that, and leave other areas untouched, or put them back into their natural condition if they are not yielding enough crop. And this is something which you can do here. And I've seen, I've talked with people on the course of my trip, and they say this is underway, it's on a good way. And for example, when you have done your Ph.D. or your postdoc here, you could go to one of these companies and develop some BT aubergines or zucchini or whatever, squash, and make people, or peas or beans, and make people more healthy. This is really important, I think.
Okay, so now to end the whole conversation, what is your advice or message to the young researchers and even undergraduates who are studying in a research environment like IISER?
An advice? I think you should try to find something which really makes you feel passionate about it. You have to have a project which you really like very much and which is convincingly interesting to you. Don't let your supervisor talk you into something. If you're not convinced, if he cannot or she cannot convince you that this is really, really important and you are not fascinated by it, don't do it. Try to find your own way. Maybe difficult, but maybe you find a supervisor who has something interesting. I mean, I did that when I did my thesis. I went to this guy and he gave me 3 options and I chose the one I thought was best. And in the course of my studies, after 1 year or so, or 2 years, I found that this was not likely to yield an interesting answer. And I stopped it and did something else. My supervisor was furious. But I did it. And I succeeded, and it was a good move because it turned out later, when other people had done things, that my initial project would have totally failed. Yeah.
So this was during your PhD?
This was my PhD. Yeah.
But you continued in the same lab, but with a different.
In the same lab with a different project, and got a PhD in this lab. Yeah.
Okay. Thank you so much for your time.